Good evening, everyone. This is what is it, 29th Journal Club for Morris Catholic. Is that the number you used, Joe? 29, something like that.
Yes, yes.
And the fourth Journal Club for Holy Cross High School, where we are going to be covering a paper that was published in Science Immunology titled Brain Sensing of Metabolic State Regulates Circulating Monocytes by the famous and infamous Joe Roman, who has his own experience. So I do want to say that I laughed out loud when I saw all of the figures, and I know you guys were bantering back and forth on the chat about it. I don't know if I've ever seen a paper that fits so many panels into a single figure. If I wrote this paper, I would have had this would have been like 15 figures, and then I would have been able to publish it. So I think that's why they just stuck it all in. So go ahead, Joe.
Okay, so it's brain sensing of metabolic state regulates circulating monocytes. So changes in energy availability, also dynamics circulating immune cells. Existing views of these affects the alter nutrient levels of affecting peripheral tissues. So basically saying that the brain, there's two different types of neurons that regulate the hunger versus the satiety of organisms. So it's saying that sorry. So that there's one type of cell that AGRP neurons. So they're focused on regulating hunger. So they're supposed to remove the amount of white blood cells whenever someone's fasting or whatever. So it kind of degrades their immune system, the vitality of it. And then there's the POMC neurons. They regulate the aspect of eating food, like getting a full, getting sated more. So they're focused more on if so they basically help your immune system get boosted if organism needs to eat more food. So in this experiment, they did this on mice. This is just the pathway of for the humans, satiety pathway. So it's just saying that basically there's just one side of the brain. I don't know if that's right. One of the figures said one side of the brain has the POMC neurons, it has AGRP. Maybe that's just a diagram. But this is just showing that the POMC neurons are on the left, how it's enhancing the appetite of a creature, and then the AGRP ones be on the right. So the POMC would be the neuropeptides, and the AGRP would be melanocortins. And then
Yeah,
so in the for figure one, first figure they show this is both sides of the brain. I think just showing it just to prove the point. At AGRP, there's a low nutrient availability, and then the POMC there's high. And then figure B, they had two mice. Or they had two groups, two control groups, and the one they injected with the AGRP drug, and then the other one was just a normal mice, I think, or the CNO drug. Sorry, and then showing that the one they stopped it, and then where is it? Unnecessarily zoom in.
let me can I interrupt?
Yeah,
Um I know we do I want to finish on time, okay?
yeah,
But
yeah,
you're going real fast because there's
yeah.
there's
Sorry.
there's a couple interesting things that will, I hope, maybe make the other students like care about this. So if you can go back to the abstract. Okay, so first thing I want to point out is that look at how they wrote the abstract, my freshman. You just got done, most of you, handing in a lab, right? That you worked so diligently on, some of you. And look at the way they wrote the abstract. It is almost exactly like that Nature summary paragraph format, right? This is how they wanted an abstract to look, or a summary to look. So you can see it says things like here using mice, here we show, right? That was one of the things for you guys. So I wanted to point that out. Next thing I wanted to point out was that we're talking about the brain and metabolic states. And Juliana, you said that this was about circadian rhythms, right? So if we go to, Joe, go to the next slide. The circadian clock is located in the hypothalamus right next to where this yellow-orange square is. So Joe, you said that it was one side of the brain versus the other. It's on both
Okay,
sides,
okay,
but they project across to the other side. So like the left side will then project to the right side, and the right side will project to the left side. And you sort of
okay.
see that with the red lines between the melanocortins and NPY. Um, really interesting to me, and maybe Maggie, but Amber Wojcinski's paper that she is writing for that independent study she's doing for me is about enkephalin, which is one of those endogenous morphines in your body that kind of relieves pain. And it is related to another endogenous morphine called dynorphin and another one that's called preopiomelanocortin or PMOC, POMC, which you can see in the upper right-hand corner of this figure, POMC. And POMC is they are the neurons that will then, if you follow the arrows, like um, Joe, stop sharing your screen for a second so that I can share mine, if you don't mind, because I want to show you guys. Let's do that one. Share. Okay. So, you guys can see my mouse, right? Yes. I can't see you, so I need somebody to give me a verbal.
We can.
Okay. Having trouble. Boop. There we go. Make this bigger. Make this bigger so I can sort of see you guys. Good. Okay. What is this? Why did that just get out of here? All right. So this is if you have ever been interested, this is a physic from my physiology course. If you've ever been interested in like how food works, why do you want food in the first place? What are the results of eating? What's the results if you don't eat? All of it's contained in this, what looks like kind of a confusing figure. Knowing Having the confidence to slow down and actually try to figure this out will help you a lot on Saturday when you're going to see a lot of these. Okay. So start up in the upper left-hand corner with the meal. All right, I eat a meal. What does that do? Well, it increases glucose. That's one thing it does. It also increases fat, right, that you're eating. Okay, so I've eaten the fat. Well, what does that do? That causes a release of something called cholecystokinin. That causes your gallbladder to squeeze out all of its bile, which bile is a detergent. It acts like soap to break up fat, the same way if you use Dawn to wipe grease or clean your hands from grease or oil, right? It suds up and it coats the fat and makes it absorbable. Okay? And when it gets absorbed, notice the positive line here, the green line, it hits the satiety center. Satiety means, I am sated. I am full. I feel good. I no longer am hungry. Got it? So the satiety center or the satiety pathway is how do you get to the point where you feel hungry? Notice this is so important in modern life, modern American life. Notice there is a direct line from fat to feeling full. There is no such direct line for sugar, which is why when you eat sugary foods, it doesn't make you feel full. You could eat a whole cake and still be like, why am I still hungry? And that's what people with obesity, that's one of the problems they have. And the new drugs for it, semaglutide and Ozempic and stuff, actually works on this center. And I bring up Amber, how interesting it is to me to think that satiety is controlled in part if you follow this, right? So glucose increases, which causes insulin. Insulin will cause you to have body fat stores increasing, right? Insulin will cause you to become fatter. Then that will increase leptin, which will activate POMC neurons to release melanocortin, which will then, if you follow the pathway, lead to satiety. As if it's as if it's like a reward. Okay, I'm going to, let's say that you've got hunger. Hunger's really uncomfortable, right? Those of you who have hunger ever, I don't. I like never get hungry. But you can't tell from the obesity that I have. The When you're hungry, it's not comfortable. Okay, so how do you relieve it? Well, you use melanocortin. All right, so what? Well, I just told you, Amber and I were reading papers today. Melanocortin is a type of endorphin. It's a type of painkiller. So no wonder you actually not only do you relieve the hunger, which is painful, but you also feel good about it. You're almost high, right? It's also one of the ones I think that's involved in runner's high. So if you go, exercise or whatever and you feel high afterwards, which only increases as you age, by the way, like you get totally wrecked on endorphins when you go for a run and you're 40 years old. You won't be able to walk the next day, but that's one way to get your drug fix. But th to me, that's pretty cool. I might actually have her put that in the paper because you are relieving the discomfort of hunger using a morphine like. And for those of you who don't, do you guys know what morphine is? I'm talking about it as if you know what it is. Morphine is probably the most famous painkiller in the world, most effective and famous painkiller in the world. It's used in all hospitals. It's in the same class of drugs, opiates, as fentanyl and oxycodone, which is Percocet, hydrocodone, which is Vicodin, et cetera, et cetera. Okay. So I just thought you would find that cool. And that's kind of what they're manipulating here. So if you go to the next figure here, Figure B is actually showing that they knocked in, which means they genetically manipulated and put in another gene so that it would respond to the drug that Joe was mentioning. Oh, hey, nice of you to join us, Lila.
That's my bad. I went through a lot of effort to come here. You better be like super grateful. You
Wait,
better
wait, wait,
reimburse me for that Uber I took. Yeah.
wait,
Yeah.
are you not home? You're like in a
No.
hotel? Where are you?
Yeah.
Where
Don't
are
worry
you?
about it. Huh?
Where are you? Where'd you go for
I've
a
been
Like
I've been
vacation?
undisclosed location.
what?
Undisclosed location?
He, I'm
It's
actually
you
getting kidnapped right now, but he like let me join this, and then when I get off, I'm
it's
gonna
good.
get murdered or something. I don't know.
He's got priorities. That's good.
Yeah.
Um,
No.
okay, so here's another interesting thing for you guys. Who likes adorable puppies? Okay. Do you ever notice that the cutest puppies have patches and multiple colors? Have you ever noticed that the sweetest puppies and even adult dogs have patches and multiple colors? Have you ever noticed that?
Yeah.
Yes, nod your head because, yeah, that's like an obvious thing. You will not meet a dog that has patches and pointy ears. They will all have floppy ears. And there is this theory about the domestication of dogs that certain neurons that go to make up the brain also make up skin cells and their coloration. And so when you have this mutation that leads to those skin cells not developing to adulthood as fast as they should, you end up with dogs that stay in their juvenile state. If you've ever seen my two dogs, from when they both passed away, but my two toy poodles that I had socks and patches all over, and they were super sweet. And we think that's actually a real thing. Neural crest cells seem to have malfunctioned, and therefore the dog does not ever get to an adult wolf state. They stay forever cute and small and less aggressive and happy to see their owners and stuff. Why am I bringing that up? Because AGRP here is actually a goody. Joe, help me out. A goody, what's RP? Something peptide. A goody related peptide. What's a goody? A goody. It's this color. So this is showing the animal known as an agouti, but that coloration is actually the color of mice and stuff. And it's called agouti. We actually refer to them as agouti mice. And it turns out that the coloration that comes from melanin in skin cells is related to a whole bunch of neural functions as well. So do you see the puppy connection here? I'm trying to make you guys care. Yeah, cool. Oh, the
I care a
enthusiasm.
lot.
This
Yeah.
is why I need Lila as an opening act here, and you guys are like dead. What? All right.
I could
Joe,
like make up stuff. I haven't read it, but I could start
We're gonna
lying.
do a Lila
Let's start
intermission.
saying random things.
Did you
Tyler,
say
tell us your story.
story?
Tyler
Yeah.
is
Me?
Oh no, save that for later. We,
All right,
we gotta
later.
we gotta finish
Oh.
on time. We gotta finish on. Joe, go ahead and share your thing again. And I'll shut up. I'll shut up. I'll stop. Sorry. And feel free to call on people, Joe, for some of these figures that you think that they can handle.
Okay, well, if you guys see it. Okay. Yeah, so figure
Yes.
one continued. They basically showed that they modified the gene for the AGRP neurons in mice they already fed. So normally the AGRP would be for mice that have not eaten all day, so that it makes them, that's the reaction in their neurons, they feel fasted. So they wanted to see if mice already were full of food, if they could feel fasted by just modifying the gene. So I think E, it just shows the concentration of the monocytes. I think it was greater in the mice that was modified with AGRP than compared to the normal mice, which had no food. So I think it's showing that they can actually increase the feeling of hunger in the modified mice than compared to the mice that are already fed that have no food. So basically, they can feel more hungry than mice that actually aren't eating anything at all.
Okay, stop. Other people. Make a connection here. Why is that? Why is that idea ridiculous? Like, why is that something that will change the world just by itself? Joe, can you say it again? Just what they're trying to do
Yeah,
there.
they're just trying to say that the connection between actual nutrition and the brain's reception of the nutrition. There's no actual correlation. It's actually just in the AGRP neurons. So if you can modify them, the actual amount of food doesn't matter whatsoever.
Okay, so someone
Massive.
else make the connection. Why is that cool?
You can well, I don't know if I'm gonna say it right. You can use the neuron more to satiate hunger
For what purpose? Yes, for what purpose?
for like kind of like Wegovy
Yes,
yeah weight
yeah.
weight
Weight
loss.
loss. Yeah. Imagine that. There are not going to be no more fat people in the world
Part two.
except me, I guess. Lila.
Um, I'm curious if this would be helpful to, what's the disorder you brought up? I forgot whether this was on the meeting with the expert we had on, or if this was another journal club. It was the disorder that where they like were always hungry, or it was. Do you know what I'm talking about? Like,
Yeah, it's called Prader-Willi syndrome.
yeah, would that help that, or like, I don't.
That
Yeah.
we think that's actually the problem. We think they're. I believe in Prader-Willi brains. I think that they have this thing called a brain bank. It's actually up at Harvard, MIT, and this brain bank stores brains from anyone who donates their body to science. Okay, and if they have certain disorders, those brains are then sliced up, and they try to see are there any anatomical or neurological or neuropeptide gene
I love
abnormalities in the
that.
brain. And what they found was that in Prader-Willi, I think they were missing the POMC neuron, something like that. They didn't make POMC correctly. So imagine you can target that. You could you know what? You don't need food anymore to feel sated. We can just give you this drug and it will directly modify just that pathway and nothing else.
How
When was
Wow,
expensive
this
would
Charlie,
that?
that's
paper
cool.
published?
What did you say?
When was this paper published again?
This is
Which one?
2025. I
Yeah,
think.
this is new.
It's
Yeah,
only like 3 weeks ago.
yeah.
Yeah.
Where, where was this? And somebody else asked a question. What's up? Who is that
I
other
was,
person?
I
Oh,
was just curious, how expensive would that be though? Because I would assume this isn't going to be like a super widespread cure for a hot minute. I assume this would be like the same as like Ozempic. I don't know, like I don't like something where it's like
I think
only
it's gonna be
accessible to
the
people,
same as
only accessible.
Ozempic.
Sorry.
I think it'd be like same kind of thing as Ozempic.
Well, because I just feel like it would probably only be accessible to people that don't even necessarily need it. Just it would be people that are able to access foods that are good for them and like wouldn't make them fat. I don't know. That's like I don't know nutrition, but
I think it is Ozempic. I think essentially that's how it works. Like
so
Joe, go back to the previous slide. Um, Ozempic is glucagon like is a GLP-1, glucagon like protein 1. Glucagon is the other thing the pancreas makes in addition to insulin. So if you're giving an artificial glucagon, you are one step upstream of this process. So if you look at where insulin is and leptin, you add glucagon to that list. Glucagon is the thing doing is the thing that uses those body stores to upregulate leptin. So it's actually doing that. It's already that's why I don't get on bandwagons. This will probably be the second or third time I talk about it, but I don't get on drug bandwagons, okay? Except for cocaine. Let's be honest, cocaine's really interesting, right? But there's a hot drug that comes out and there's always bad news about it. So, there's people end up having weird, terrible side effects. It doesn't work the way it's supposed to, blah, blah, blah. This semaglutide's been around for a while, and there are very few side effects, and people are having amazing results with it. We're not going to have fat people anymore, except me, obviously. Yeah.
It's
All right. Sorry.
conquered obesity.
Yeah. Yeah. All right. So, Joe, be judicious here about which ones you think are important to cover.
Okay, let me just do this. I guess figure one, they just like prolong the experiment by like actually fasting the mice. So they basically just like showing that even if they don't give them, if they actually make them hungry on purpose instead of just not feeding them, they actually prolong it. I think they said it was like they did something like 16 hours of fasting, and the effects were still prominent. That the gene modification actually had a greater impact than the mice who actually were fasting. So it just proves that the impact more.
Use
And
your
then
mouse to point to the things you're talking about for us, okay?
okay, I 1 minute. I'll use the mouse. Okay. And then the fasting, and then I guess the glucose levels were very similar. I think they were even lower in they were slightly lower, like in the K in the first diagram. In the AGRP neuron modification, they were slightly lower than the fasted mice. So it shows that you can actually decrease the glucose level just the drug. And then L is just the activity of the neurons, and it's just showing that the activity it's about the same as having a fasted mice, actually compared to like a mice that actually was refed and then had the neuron modification. So just showing that it just proves it again, and then I guess M and N is just a scatter plot of L. Okay, figure 2. Who should I nominate for this?
It's the number of monocytes, right?
Yes.
So you're seeing, and I'm looking at a black and white copy. It's not showing up as well. Can you go back up to 1? And so it's saying, see the stars, guys? They're right. The stars mean significance. So meaning P is less than 0.05. So that is saying there is a significant difference between the fasted and the refed. And there is a difference between the refed with the activation and the regular refed, but there is no difference between fasted and the refed with the activation, which is good, meaning that they have accomplished what Joe just said. They have accomplished a model of fasting without the mice having to fast. And that has a bunch of profound implications for other systems. Like if you have bipolar disorder, for example, I wonder fasting tends to help with bipolar disorder. I wonder if you can use this method to then reduce the need to actually fast
How
and
does that help?
still have the results. All right, go ahead, Joe.
Okay, yeah. So I'm going to figure 2. This was what was it here? I think they went into they dove into the other pathways besides just the neuron modification itself. Well, let's go back to the paper 1 minute. Can you share your screen, Char, or should I just leave it up? Okay, I want to look at the paper real quick. I guess I can just skip over that. Where was it? What's
Is
happening?
there a way to share just a tab on Zoom? I know Google
They're away. 1 minute.
Google whatever Meet has that, I think. I don't know if Zoom has that.
It's the paper here. Sorry, 1 minute.
Sorry, small side note while he's looking for that. I just have a question. This might be like something you kind of already explained, but I just kind of want you to spell it out because I don't really know the answer. We were talking about this versus Ozempic. What makes Ozempic bad and have all those negative side effects versus what we're looking at now, like in this paper? I know it might be like a really. Are you muted? I just can't hear you.
Sorry, I didn't hear what you said. Don't. What makes Ozempic what?
Like have all these negative side effects, like be bad. Is it the addiction that makes it bad for people? I don't.
Ozempic?
We were talking about Ozempic before, were we not?
Yeah,
Or no?
but the
Not bad.
the reason I am actually jumping on the Ozempic semaglutide bandwagon is because while it does have a few side effects, they're so negative, they're so minor that it's an extraordinarily safe drug. Like way better than I would have expected. So
Okay.
yeah. But
I
I know more than probably a dozen people on it right now who, and all of them are like, yeah, you get a little bit of nausea on day 2 or 3, and then it goes away, and I'm just not that hungry. And then they're losing weight like crazy. The bad thing is they also lose muscle mass, so you do need to exercise. But like amazing results on it.
okay. I
Yeah.
don't know why I thought there was some sort of negative stigma behind Ozempic. Maybe I'm
Yeah.
thinking of
Well,
something
there's
else.
going to be the gym bros who are like, no, man, you got to do it yourself. You can't use drugs to do it.
People will call you lazy, I think.
Where's Cameron Wright when
Right.
you need him? He would absolutely eat this up, unfortunately.
Yeah. Where are those guys?
Uh, no idea. Probably vacationing away. I don't know.
If
Cameron's
I'd known
probably
you guys
eating.
were on vacation, I wouldn't have done this. I would have done it the
Are we?
following.
We're
I
all
didn't
on
realize
vacation,
that.
right? I think we all are.
Yeah, I didn't
I
realize
love being
that.
on vacation and doing this.
Ours
Like
ours
Ryan,
were
like
last
the
week.
view.
Yeah.
Colin's so fake for just saying whoops and then not saying anything else.
Just
Like
no.
I said whoops and then I tipped my driver $5 even though it was a $9 drive. It was great.
You're
He was very
with
nice too.
Colin?
Huh? What?
You're with
I did
Colin?
not say that.
No, he said driver, Uber driver.
Driver.
No, no,
What?
no. You said Colin said whoops, and I tipped
In
my
the
drive.
no
Oh,
no the Google
And
chat
there's
he
video evidence of this, so I don't know
no
what
no
you're
no
talking
okay
about.
I phrased this wrong.
oh,
You're
okay.
like being very ableist right now. No, on Google chat he said whoops and that was all he said. Unrelated. He said that, but see I like went above and beyond.
Mm,
Yeah. Anyways,
mhm.
sorry. I'm sure Joe thinks ready.
All right, so
Let's go back to the presentation. Yeah.
this
So
one.
figure 2. Yeah, that was it. Yes, they're basically this one. So for A, they basically they have an experiment timeline. So
Mhm.
they wanted to see that the neuron activity is actually the only thing that's required. That it's like the main thing that regulates the monocytes. So they wanted to figure out if it was actually if they like silence the neuron activity, if that would actually change the monocytes. So I think in on B, that's the TC expression, which is some light expression which blocks the neurons. They said there was no difference in the body weight and the food intake between the control and the one they did with the TLC light expression. So basically showing that even when the neurons are turned off, that the mice when they're turned off, the mice see no difference between the control and the normal ones. So it suggests that the neuron activity is the only thing it's the primary requirement that it's the one thing that regulates the monocytes.
Okay, so why is that important though?
I think this is just to show that they actually established a causal basis for the monocyte regulation because they're basically saying that no other factors contribute to the like if they can't really modify any other aspects of like the amino pathway instead of neuron activity because neuron activity is like the main it's
Yeah.
what causes it. I don't know if that's what they're trying to get to.
Well, so let me if you don't mind, I'm going to give you
Yeah, yeah.
a slight criticism. So
Okay, sorry.
in
Yeah,
this no, that's fine. It's in a presentation like this, guys, you do want an introduction.
yeah. I should have done that. I was
Yeah.
gonna like okay.
Because what he's saying is also really interesting because We didn't know this. There was some hint that the brain was controlling it, but people actually thought that it had to do with changes in the immune system and changes in actual calorie amounts that are available. That how much energy you had in calories from your food would actually change your immune response. That's what's been thought for the last probably 100 years. And now, as Joe just said, they're actually showing that the vast majority of it is actually controlled by the brain. And if you told me 20 years ago that the brain was controlling the immune system, I would have thought you were crazy. That is a new, really cool thing. Because you can now maybe manipulate brain stuff to manipulate your immune system instead of having to do ignoring the brain and trying to do other methods. Those, because you've all had infections, you've all gotten boo-boos, you've all had, let's say you've had an injury or a cut or something that gets infected and you have pus and stuff in it. The pus is made of lymphocytes and monocytes. And so they are fighting the good fight to get you healthy again and make sure you don't die from that paper cut. But it turns out you could actually get them to upregulate or downregulate just by manipulating the brain instead. That's pretty cool, right? If I want to, you've probably, how many of you, you don't have to admit this, but you might go, oh, yeah, that's me. How many of you have been on prednisone? Right? Prednisone is a steroid. I guess it's just the three of us, Kara and Dylan. But prednisone's a steroid, so Dylan's been doing roids. But it's a steroid that actually tamps down your immune system, and it has tons, way more bad side effects than Ozempic, for example, Lila. It's like it's a great drug. It's very important, but it just really screws up your body. What if we could do the same thing just by manipulating the brain instead? That's why it's cool. Okay? Everyone, stop being bored. Caden, stop being bored. Yeah. All right, go ahead. Call on somebody.
Okay, Tyler, you wanna finish this one?
Who?
Tyler.
Tyler. Oh, they said Talia.
Sorry.
I was like, who is Talia?
What? Or are we on?
Or do you want to do the fourth thing?
Oh,
We should nominate you last.
Yeah.
Who can I call in then? One of your students, Dr. Char, who should I, participants.
Come on, people, don't make him call on somebody. Which panel are we on?
We're on figure two.
But what panel? We're on.
Oh, sorry. We're on
D.
D. Yes.
Yeah. Oh, one last thing because I can't help myself. When you were talking about shining a light on it, that experiment, it's a gene that's connected to an opsin like in your eye that responds to light and then turns the cell on and makes it fire. That is Carl Diceroth did that with well, it's mostly Carl Diceroth, who's at Stanford. He will win the Nobel Prize for that in the next probably 10 years. It is you'll probably see it on Saturday, somebody doing that experiment. It's very, very cool what he was able to create, where I can shine a light on a neuron and all of a sudden it'll start firing, even though it's not light receptive. So that's what that TETLC is. All right, so D.
Should I just continue or should I nominate? So I guess there's so
I can just try to guess.
Excellent.
yes, yeah.
I will feel to tie it back as well, but like I don't know.
You haven't read the paper. I don't know. Good luck.
Yeah, exactly. Well, I'm not gonna be able to tie back to anything important, but I think D and E are. But I think D is about measuring the weight, so it's saying like the body weight stayed normal even when they turned off the like AGRP neurons. I think that's what they're trying to say. Maybe I don't know if there's the actual conclusion of the paper, but I know that. Sorry.
D's actually not. D's a negative control. It's actually a control. It's showing that the GFP thing doesn't do what the TLC does, and therefore you're not gonna see a difference. That's it's just showing actually a control. That's all. So it's actually not that interesting. I think you want to skip to probably G.
Oh, okay. I had a little when I saw E, I was like, oh, that's like a sound, whatever. G.
They didn't label this very well, did they?
it's just measured weird and all the abbreviations I haven't like Googled yet.
Okay, hold on, wait. My kids. Look at G and J and L. What did they do wrong in the figure?
Did they not explain what the different colors mean?
Exactly. Now, did they? They did in D. And I think that they just assumed we would use D as the key. But I'm not actually even sure. In the interest of time, though, ultimately, what are you seeing?
I guess we just see there's no difference if you turn off the neurons. It's just basically just the same as the control. So it's showing that if you turn them off, that they're not going to regulate it anymore, and it's just going to go back to normal. I'm guessing, yeah, and that's most of this. But it's just saying about the same, like for H, it's the same percentage of the monocytes, I think, in the control versus the one with the TELC, TTLC. And then it's showing that in I, it's showing that the intact neurons, that the activity is much different than than when compared to the, or I think, wait, yeah, it's compared to the
Yes.
monocytes. It's much differing activity. When they disable it, it's the same activity as a normal facet mouse, I think.
Yeah, but look at the x-axis. Anybody have any idea what the x-axis means for I? Juliana? Okay, if I'm calling on you, Juliana, take a guess what it's about.
Something about the neurons.
If I'm calling on you specifically, it's because,
I'm supposed to know it.
well, because you are the one who told me.
Oh, about the circuit.
Yeah, it's circadian.
Oh.
Yes, it's circadian. ZT means Zeitgeber time in German. So ZT Zeitgeber time is what time of day relative to the circadian clock. And what happens is these neurons are right next to the clock. The clock will actually up or down regulate them on a normal rhythm, right? But in the right panel, the disabled neurons are not actually cycling correctly, which is cool. That means that it's actually no longer as receptive to circadian input. So that's a whole other field where they can jump off and write a whole bunch of other grants and papers for. Cool. Let's go to 3. And hold on, I just want to be really, really discerning here because I want to get out. Yeah, this one, I started to wonder, did you pick this paper because of the liver thing, Joe?
No, I just picked it because of the just like the kind of the hunger thing. I didn't really look at the like not really the liver part mostly.
I thought you were trying to save my life or something.
Yeah, I
So
could
never
have
mind.
known
Screw you, Joe. I don't need your help. So neuron liver axis. So the liver is one of the most circadian organs in the body. It has its own rhythm also, though it's under the control of the brain. Well, I'm having a hard time understanding myself. I got kind of stuck there myself.
So I think they were just showing that the liver activity. They wanted to compare the liver activity to the brain activity to see if like if they wanted to see if the liver regulated it played a greater role in regulating the immune system than the brain did, I think. So I think they were looking into the liver of the mice. They injected they injected something into the liver axis. So they're just showing that if they injected it that there was or no, wait.
I think they're
Or
just
maybe it's just
trying.
the brain. Is it just reiterating what they said in like the figure 1?
Yeah, so remember when I said there's this thing called an HPA axis, hypothalamus pituitary adrenal gland. Right, Lila, you remember this from phys, right? The HPA is your stress, right? Controls, right? And there's also a hypothalamus connection to the liver, where it's going to be the brain, the homeostasis in the brain is trying to control how the liver is doing its job, but the liver's job is to filter the blood of any like toxins and stuff, but also to, I guess, I didn't really know this, but I guess chain control those monocytes. That was news to me. I didn't know it did that.
So they're
So
saying,
maybe skip it.
yeah, I guess skip this one.
Yeah. Yeah, because I don't quite understand what they're trying to get at
Yeah,
there.
I mean, I guess maybe they're just saying that the neurons in the brain like regulate the liver, and then the
Mhm.
liver also regulates the immune system like through the same way the brain does. Maybe they're saying the brain controls like I don't know, but yeah, we'll go to figure 4 then. Can you do it on your screen, Dr. Char?
Oh, I'm sorry.
Oh, it's okay.
Sorry. Yeah, go ahead.
Okay.
And guys, if any of you need to leave for some reason, you can leave. I told you guys we're going to try to keep it to 8, and I can't help myself. This one's about glucocorticoids. So this is the HPA stress response. So I don't know. I should really talk to Miss Flaminio and see what she covers with you guys in health. Did you guys do stress and health? You did. Okay, and she probably told you that when you're stressed out, you can get sick. You have a better chance of getting sick because the stress hormones, cortisol, cortisol's designed not as the stress hormone to stress you out, but as the hormone that your body uses to relieve stress so that you can deal with whatever's stressing you out. Now, what's supposed to be stressing you out is not, oh my God, Johnny broke up with me. It's not supposed to be that. It's supposed to be, oh my God, there's a saber-toothed tiger trying to eat me. And so, if a saber-toothed tiger's about to eat you, do you really need to worry about the boo-boo that you have and the pus in the boo-boo that you have on your paper cut? Do you need to worry about that? No, because you're about to be dinner. So, what court does is it actually turns off your immune system. This is why prednisone, I mentioned prednisone earlier, that's what it does. Tamps down your immune system so that you can use your energy and other resources to get away from the saber-toothed cat, right? Or to fight back, fight or flight. Which actually, that might have been the point of figure 3, because the liver is involved in the parasympathetic regulation. But I still don't understand it. So, okay. What's the kicker here? Like in one panel.
I think because they said there's a synergic relationship, so I think they're saying that the neurons, the glucocorticoids without the neuron involvement, they don't really have the same impact on the hunger level in the mice. So I think they're saying that if the neurons and the glucocorticoids work together, they have a much greater. They actually implement a change in the hunger at the stress level of the mice. So just showing that the neurons have a greater dominance over the control than the glucocorticoids do, I'm guessing.
Yeah. So we're kind of skipping here, in the interest of time, we're skipping the panels, but in getting to kind of the conclusion of it, which is very cool. Because I said, and I'm glad, Lila, I'm glad you said, and you may not have meant this, but that there were these side effects for Ozempic and it was dangerous. And then I compared it, which I don't think so, and then I compared it to prednisone. Prednisone is used all the time for lots of different things, and it is dangerous. It's if you take it for too long, you start having Cushing syndrome and a bunch of other problems. Wouldn't it be nice if there was a way to have the same effect as the steroid by instead small doses of steroid and activating this part of your brain? Then you could avoid a lot of I'll use myself as an example. The liver thing I've been having lately, the treatment for it is a very high and long-term dose of steroids, and we've avoided that because I'm already fat. I don't need to get fatter. and also a bunch of other problems that it would cause. But if instead you could give me a low dose of it to tamp down the inflammation in my liver, and just by activating parts of my brain, then I get all the good effects without the bad side effects. So that's like super useful.
You could be Mr. Olympia, man. Get this.
Right. Well, I mean, so oddly, the court doesn't really affect muscle growth. I'm not AJ or anything. But yeah, one person got that joke. So yeah, why don't we go to let's say O is kind of the model here, right? So it's saying that AGRP is causing an upregulation and thumbs up, I guess. That's a weird symbol of CORT, which is the stress hormone. But it's also blocking this thing in the paraventricular nucleus, which itself would have signaled to the liver, which would have signaled to, oh, that's the connection. Oh, and notice the question mark. Remember I said it's news to me? Like I didn't know about that. Well, there's a reason I didn't know about it. They don't know about it either, hence the question mark. It's conjecture, I think. So liver activates bone marrow. Bone marrow is where you grow white blood cells and red blood cells, right? So that will then decrease your plasma by increasing your cells. And so you'll get you'll get a change in the number of monocytes. That's the model. Cool. Okay. Conclusions. Joe.
Oh yeah, so in conclusion, so we're just saying that the neurons, both the AGRP and the POMC, if we modify either one, we can change the feelings of hunger and satiety in the immune system of like mammals, like mice or maybe even humans. I don't know if they have any like human tests on this yet because it's such a new paper, but that's what they're mostly just saying, that there is that neural circuitry really controls the feelings of hunger and satiety. And then I guess we're saying that if we can regulate the immune system through this neuron reception modification, we can lead to immunomodulatory benefits without the need of nutrient addition. So I don't know if this could be people suffering from like, I don't know, like maybe like malnutrition or something. Maybe they have an illness in the hospital. Maybe they're unable to eat something. I don't know. Maybe
Yeah,
we
cancer,
could, yeah.
chemo. People on chemo, one of the drugs that they take to counteract some of the side effects of the chemo are long-term high-dose steroids, which then causes a whole bunch of other problems themselves.
Hmm.
Cool. Okay,
Yeah,
I gotta, you guys, it's
I
gotta
don't
be,
know. I wasn't very
yeah, it's
fun
gotta
with
be a
this.
new word you've never heard of, not just like
Because last time we
hunger.
were joking with the hair one, I don't know. Because I guess that's just because of a different paper.
Does the paper belong in
That
science
was Dr.
immunology?
Char reference.
Yeah, that's why. Yeah, I had to reference someone else. There's no one to reference for this.
Does the paper belong in science immunology? With an impact factor 17.6 is high. That's a pretty good journal. Does it belong in
Yeah,
a
I would say yes, then.
I think so too.
Yeah.
I think this is a pretty good paper. Yeah. I wish I understood it a little more,
Hmm.
but it's
Maybe if they put like figure O like earlier, or oh, sorry, O on figure 4 earlier in the paper, maybe like during the liver when they talk about it, maybe
right. So
understand it
so
more.
that's a suggestion I might make is just because it's in the last figure doesn't mean you as a presenter have to show it last.
Yeah, yeah.
You could actually take it yourself, stick it up front and be like, look, I'm not trying to lead you down a long story because you're a scientist and your time matters. And I'm just going to tell you right up front, here's what we think is going on. And there are going to be people they'll do this at the poster session on Saturday for my guys, where they'll tell you, look, here's what we conclude. Now, would you like to continue? And I can show you why. And so the people who are really interested in it will follow the rest of the narrative. But a lot of times we just don't have the time or interest. So we just go, no, no, I'm good. Thank you. And then you go to the next, which I find kind of rude, but then you go to the next one, the next poster. Okay. Is there anything else? No, that was it.
I think that's
Yeah. Any questions, comments, concerns? Sorry about the rush, Joe, but good job.
not my
Thanks for
fault
doing
on
this.
joining too late. I still the webcam still doesn't work. I'll figure it out eventually.
That's right. So I don't think we're going to have another journal club next week because we are going to the Met.